Thank you for joining us for the final talk of this year's Innovation in Disease Modeling Symposium. My name is Princess Senit, conference producer at Technology Networks, and I am here to introduce our final talk of the day, Cardioids Unravel Human Heart Development and Disease. I am pleased to welcome Dr. Sasha Mendjan. Dr. Mendjan currently serves as the principal investigator at the Institute for Molecular Biotechnology in Vienna, where his groundbreaking research delves into the intricacies of human heart development, regeneration, and disease mechanisms. A warm welcome to you, Dr. Mendjan. Following the presentation, we will have a Q&A session, and we welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. I will now hand over to Dr. Mendjan to begin his presentation.
Thank you, Princess, for the kind introduction, and also for Technology Networks to give me the opportunity to present our work. I will talk about how we use cardioids to study early human heart development and also cardiac defects and disease. Now globally, by far most people die because of cardiovascular disease. You probably know this. You see the number is about 32%. People die, but it's like every year it's actually the same. So 2018 is not an exception, it's rather the rule. It's way more than, for example, cancer. But the real problem that we have is that new therapies are extremely rare in the cardiovascular area. So about just 2% of new therapy breakthroughs are actually in the cardiovascular area, while, for example, in the cancer drug area, this is much more wide. About 45%, and this is again for this period of time, 2013 to 2018, but it didn't change actually over last decade. Actually, in fact, it didn't change in the last 30 years, which is a huge problem. Now, the major bottleneck that we have, why this is so, is that we lack physiological models of the human heart. And what we mean by that is that the current models are not really predictive enough at the preclinical stage, or when we want to, after we want to develop therapies. So they're not really telling us, how a particular drug will work. Also for drug development itself, we don't have, the models are actually not good enough. And that is a huge problem, especially in the cardiovascular field, because their clinical trials are extremely expensive and extremely lengthy. And this is one of the major reasons why you see this problematic statistics that we have. Now, the same issue that prevents us from understanding how basically cardiac disease actually comes about and how we can treat also prevents us to understand cardiac malformations, so cardiac defects in babies. They are really difficult to study and to prevent. About 2% of all newborns will have a cardiac defect. And for example, here you have one of the most prominent one, hypoplastic left heart syndrome, which is a defect of the left ventricular chamber. And that is completely obliterated, as you can see here in this. So what this essentially means that a lot of these defects are also compartment specific, that they will affect either, for example, only the septa or the the ventricular chamber, the right ventricular chamber, or the aortic arch, etc. And again, we don't have really models that are good enough to understand that, specifically not in the human. Now, why is that so? If you look when cardiac development actually starts, it's the first organ to develop. Already at day 19 in the human embryo that looks like this, it's essentially a disc. You have already these cardiac progenitors here of the left ventricular chamber, of the right ventricular chamber, of the atria that are exiting the primitive streak. So a structure from where cells essentially start to differentiate and to form these cardiac progenitors. And they already determined at this early stage. So day 19, which is incredible, right? It's before before the women even know that they are pregnant. Now, these progenitors will, within a few days, here, day 25, they will undergo a very intricate process of tube formation. So they will start forming the really early primordial heart that is mainly made of these left ventricular progenitors. And this heart will keep on adding progenitors, so-called second heart field progenitors, that will be added on both sides of the heart, what you can see here nicely. And the special thing about this stage is that the heart already becomes functional. So it needs to start really pumping and establishing the embryonic circulation. And then we have already something that looks much more familiar to you, probably, the fourth chamber or the chambered heart is start developing with the left ventricular chamber, the right ventricular chamber, the outflow tract structure where the big vessels will start getting out. And then, of course, also the atrial chamber here. And a bit later, then also septation will start. Also pacemakers will be added. Also, what will be added is this epicardial layer here, which is surrounding the heart, that is important for regeneration of the heart. While the myocardial layer, the major muscle, is basically in the middle, and you have the endocardial layer on the inside here of the heart. So all of this is already, you see, done within the first month or so. And all of these steps here are very difficult to study, especially in the human context, because it's basically a black box how this happens in the human. If you want to study this in animals, the additional sort of limitations are the speed of development, the complexity of all of these different progenitors, and also the dynamics. How do these different compartments interact with each other as they develop, and how they build a physiological organ that we are interested in. Now, to overcome some of these limitations, the field came up with different type of in vitro models that are mainly derived from human pluripotent stem cells. And this happened in the last about 15 years, I would say so. So one very prominent model is, of course, the 2D and the 3D cardiomyocyte differentiations that you can see here. These are these nice beating sheets of cardiomyocytes. This was very useful to understand the role of particular signaling pathways during the different stages of cardiac specification. Then you have these microtissues that are very useful to understand how different cell types, cardiomyocytes versus, for example, fibroblasts or endothelial cells, interact with each other. Then you have these engineered heart tissues that are very useful to understand the mechanical, some of the mechanical properties, because you can measure essentially things like force with them, what you can nicely see here with these pillars. And then you have also some of the embryonic models where people start to look at the context of the cardiac cells, also in the wider context of other developing organs. But this is still, this field is still in its infancy. Now, when you look at all of these models, and when you compare that to the real thing, which looks roughly like this, you see that there's still a large gap. And you also immediately then see why, why the problem that I outlined at the beginning is still so big. Because, for example, what we are missing still here is the proper cardiac morphogenesis. So, for example, the chamber, a beating chamber that is pumping actually liquid with all its mechanobiological properties that then play a huge role also in disease development. Then the different compartments, which we also didn't have, the right ventricle, left ventricle, atria, that we couldn't really model all of these. And then also the growth of this organ. We don't really understand exactly how the human heart also grows, how it proliferates, while it functions at the same time. That's not very well understood, and we can't model it in vitro. And then also we can't really remodel in vitro regeneration or repair or in general processes that also we see during myocardial infarction, for example, like fibrosis and scar formation. All of these things we were not able to model in vitro, and that was a huge bottleneck. Now, in our lab, the approach to tackle all of these issues that I just mentioned is that we start also from human pluripotent stem cells, and we generate these very specific mesodermal progenitors using signaling exactly as it would happen also during development. The goal there is to get these self-organizing structures that contain organ-specific cell types. So basically cardiomyocytes, endocardium, also epicardium here. We want to model specification and patterning to a certain extent as it would happen in vivo. So this doesn't mean that we will be, able to perfectly model it, but better than until now. And also to start modeling morphogenesis, which hasn't been done before, like, for example, this formation, for example, of the cavity, cardiac cavity, etc. Now, what's the advantage of this type of approach? It is that this is a reductionist model where you can then start dissecting mechanisms because it's simpler, of course, than the in vivo models. It's more amenable to imaging, to the genetics, you can do proteomics, and importantly, it's in high throughput. So it can be done in 96 well plates or in 384 well plates. Now, the final point I want to make here is that really, and especially also to viewers, for example, from industry, etc., that this aspect that we follow development on and via, having self-organization and development essentially in vitro is extremely important also to model disease or to have to develop models that will be capable of modeling disease as we want it. The reason is because if you look at disease, many mechanisms, if not all of them, will have a very strong developmental component, meaning many mechanisms that are already active during development of an organ will be later on sort of aberrantly deployed during disease. And that's what's basically the underlying pathology often. It's that these are developmentally guided mechanisms that are switched on at the wrong time in the wrong place. And that means that we need models that can recapitulate not just disease itself, but also development first. So that's our principle behind what we work on. Now, to get there, what we started with, and this was a couple of years ago in this study, we started with human pluripotent stem cells. We differentiate them into cardioids in high throughput, and this is done without exogenous ECM, which is very important, into these chamber-like structures. They have essentially fluid-filled, have a fluid-filled cavity, as you can see here, and that structure you see is beating, and this looks resembles much more of a cardiac chamber than some of the models before. Now, what could we do with this model? First, we could, for the first time, model a morphogenetic cavity defect. For example, particular mutations that they would affect the formation, for example, of the cavity itself, like genes implicated in the hypoplastic left heart syndrome that I mentioned before, HAND1, that was heavily impacting also the chamber formation of these structures. Second, we could go into, for example, the self-organization of cardiac lineages and how is this regulated by signaling. This was also not understood very well before, or not at all, actually. And then also for the first time, we could see an injury response, an ECM response, a rudimentary sort of scar formation by accumulation of collagen and fibroblasts at the injury site, which also hasn't been seen before. And the underlying reason there is that we did not use exogenous ECM in the whole process. And that means endogenous pathways started reacting much more physiologically than before. Now, the limitation of this approach was that we were focusing on the developing left ventricle. So this the earliest part of the heart that starts to develop, but we didn't have still models for the other compartments that I told you before. This differentiation protocol could be recapitulated using different lines. Here you see that we, in the paper that I mentioned, we did actually use mainly the H9 line and VTC IPS cell lines from the Allen Institute extensively, but we also did it in 4 additional other lines. And then now in the meantime, I think the number is probably over 15 different lines that we differentiated it. And this works well, especially it's very reproducible within a line, but between lines, you will still see some differences, but they are again reproducible. So it's easier to handle. Now, one advantage of the platform is its modularity, meaning there are different versions of the protocol where you can, for example, you can look at only cardiomyocytes. So cardioids that contain only cardiomyocytes are about 90%. You can look at cardioids that contain cardiomyocytes and endothelial cells, or cardiomyocytes, endothelial cells, so with the lining here, and also epicardial-derived fibroblasts, for example, that are start migrating in. So that's also possible to look at. And now, more recently, in the collaboration with the All-Over and Mammary Lab, we can also add macrophages to the system, which you can see here nicely start homing on the injury side, as it would happen also in vivo. Another advantage is the throughput that I already mentioned. They form reproducibly, and you can see this here, that they start developing over a time course of 7 days, all in parallel, and you can see that this can be also adapted to a 384 well plate. Now, it's not just that we can do this in high throughput, but the whole differentiation process and analysis can be automated, which is a huge advantage. Again, this is because of the absence of the exogenous ECM. There's no matrigel embedding or engineering involved. So that means that a lot of the existing robotic technology can be relatively easily adapted to start producing these. And then also, of course, software can be adapted, and different machines that can be hooked up to this type of liquid handlers that you can then start analyzing, their shape, their size, the expression levels of certain reporters, how they beat, etc. And then you can have an internal QC that will basically tell you, okay, how good is this differentiation, and that doesn't depend anymore on a human operator. So this becomes actually very much, much more objective in a way. But I told you there was a limitation because we were focusing just on this left ventricular chamber here. Why is that? The limitation is because, for example, if you want to look at congenital defects or also many other diseases of the heart, the left ventricle, especially for the congenital defects, the left ventricle actually, just 3% of the congenital defects are affecting left ventricle. Most actually will be happening in the other region. Of course, the most common ones are the septation defects. But also other regions like the outflow tract or the atria will be heavily affected. So the second generation sort of PhD students in the lab, they started developing a model that would address this problem together with Toby Ilmer in the lab, a research assistant. And what they found is that mesoderm induction and patterning signals that they already determine in the first 3.5 days of the differentiation. So these are these signals, what you can see here. They determine these early cardiac progenitors that will later then specify into the different chambers. So it's essentially, which means that really it's extremely crucial which signals and which combination of signals you use at this first 3.5 days to get then later on the right heart compartments with the right identity and also the functionality. So to give you an example, when we start this differentiation and analyze the progenitors at day 3.5, you already see here very nicely that they express specific transcription factors that are very specific for the identity of these different type of progenitors. So, for example, these ones here will be the anterior second heart field, these one for the posterior second heart field, and while these ones here are more highly expressed in the first heart field progenitors. These will then, you can see when you can look at, of course, at the RNA level, but you can also look at the protein level and quantify with the quantification here. You also see a nice difference in morphogenesis, that the left ventricular progenitors will start developing first, while the second heart field progenitors didn't start morphogenesis yet. And this is exactly also what we see in vivo. Now, if you then go further in the differentiation, here we're looking at day 14, you see that they all start forming cavities, but they will express different identity markers, like IRX1. It's much more highly expressed in the right ventricle, while NRF2, for example, will be much more highly expressed in the atria. Here you see this with the quantification. And then, of course, if you then do a single cell RNA-seq analysis and compare that not just to 1, 2, or 3 transcription factors, but really transcription factor modules and generally genes that we know are relatively specific for the compartments, you see that we were very specific how we differentiated. You see that we can distinguish nicely between the left ventricle, the right ventricle here, And then also the different regions there are, the outflow tract, ABC, and atria. Then another advantage is that we can then also start also a maturation process of these cardioids so that they mature further and while retaining identity. You see this here nicely. So MIL2, so myosin light chain 2 protein, you see that it's highly expressed in these matured cardioids here after 30 days compared to the early ones. And interestingly, also their functionality is actually improving further. So you see that upon maturation, they also beat have a stronger beat, and especially in terms of amplitude. The amplitude of beating becomes much higher. Now, this is all nice, but the question is like, okay, what can we now do with this? Now, especially in terms of looking at cardiac defects, when you look at the causes, you can have environmental causes. About 10% we know are caused by environmental factors like different drugs, thalidomide and Contagan, of course, the most famous example, which is a human-specific drug that was then called, or so human-specific sort of teratogenicity that was not really seen in animals, for example. Then, of course, retinoids, all the skin creams that, of course, can have a really devastating effect on cardiac and also in embryo development in general. Maternal diabetes, another one. Then, of course, we know about the genetic causes. About 34 have different genetic causes. Can be single de novo mutations, can be also things like chromosomal anomalies. And then you have also environmental factors like plastics, enhancer mutations, or multifactorial things that we actually cannot really test. And these are unknown. These are basically unknown. We don't know what's the cause here. And this is really a big unknown, as you can see here, despite all the screening that is happening in the hospital. And then what we were lacking until now is systems really to test the potential causes of all these cardiac defects, especially of the unknown ones. Now here I give you an example of what for example, we looked at the gene islet1 that we know has a very specific mutation in the second heart field progenitors, then also in the derivatives like the right ventricle. And then we wanted to test our platform. Okay, can we see a specific effect there? Now, if you look for islet1, for mutants, if you look at the cardioids in terms of size, you see that in the mutant, they are generally smaller, especially the atria, outflow tract are more heavily affected in terms of size. The right ventricle, not so much. The left ventricle seems to be the least affected in terms of size. But then when you do a section of these, you see the real difference. In the knockout left ventricle, you see that they are completely fine in terms of, cardiomyocyte specification. They also beat normally and everything, while the right ventricle has a total absence almost of cardiomyocytes here in green, although the cavity is still there. So, which tells you first that the platform can nicely distinguish even between the left and the right ventricle. And you can also distinguish nicely between effects that are on specification of cardiomyocytes versus morphogenesis. So here in this case, morphogenesis is not affected, but specification, for example, is. Then we looked also in terms of functionality, what happens there. When you look in the outflow tract cardioids, for example, they also show the phenotype, but the different one. There you saw upregulation of some of the atrial markers, which is interesting. But then if you look at the functional level, the outflow tract, the wild-type outflow tract doesn't beat on its own. It needs to be hooked up basically with other, with a pacemaker to be paced, which makes developmentally sense. But if you knock out islet1, you see that it starts contracting at a quite high rate. That is quite similar to the atria, which means it started changing identity. So these outflow tract cardioids really adopt atrial identity in these islet1 knockouts. Now, another example where we use this is to see, okay, can we start testing, drugs like acitretin, for example, that you would have, as I said, like in some of the skin creams treatments. And there we also saw quite remarkable effects. For example, here, if you look at this outflow tract here, when you just look at it, especially at the lower dosages here, which are like just 5 nanomolar, so it's a very low dosage, even below what's normally found in the bloodstream. you see that the outflow tract suddenly started taking on actually a ventricular identity here, while the other ones not. So you see really compartment-specific effects now, and also this outflow tract also started beating again, which was also quite remarkable. So that essentially means we can now really start looking at really, compartment-specific effects, both of drugs and of mutations. But then we asked, okay, can we, make this model even more complex and start asking questions that we couldn't do at all before? So, for example, one question is, how does the human heart even start to beat? This sounds sort of trivial, but we don't really understand that because, as I said, this stage of human cardiac development is essentially a black box. All we know actually how the heart in general starts to beat is actually mainly from this paper in Chick, where they looked at different stages, and then they did electrophysiology of the different parts of the heart to really get the idea. Now, what we see in terms of these cardioid subtypes, it's not just that they express different genes that I showed you before, but also that they have a different functionality. And different functionality is exactly what you would actually expect also in terms of in vivo. You see here that the atria region and the ABC region are beating the most, while, for example, the left ventricle less and the right ventricle outflow tract even less. We can look at these differences also by using MEAs, for example, where you can see basically how these different signals are, passing through a cardioid. And also we can look at them like the GCAM type of propagation, calcium ion channel propagation over time, which you can also measure, where you can measure the speed, etc. Now, these cardioids, they can be also put together at a very specific stage, at the early progenitor stage, and led to co-develop. Now, if they co-develop, then they will form what we call here multi-chamber cardioid. This is a structure that shares a lumen, what you can see here nicely, but has different identities in the different sections. And they, of course, started interacting. Then we asked the question, okay, can we now, if you look at this structure over time, can we understand how the human heart actually starts to beat? And then on day 6 of development of this type of multi-chamber cardioid, what you see is that the beat will start from the left ventricular chamber here. And this is exactly what the people saw in the chick. So now we have a more human model that shows actually a very similar type of pattern in terms of functionality. Now at day 9, this changes. The signals will start from the atrium, go over the left ventricle and ends in the right ventricle. This is exactly the flow that you want to have, because the blood will enter the atrial region, will go to the left ventricle and go into the right ventricle. At this stage of development, you don't have yet the septa. So this will happen later. And of course, with this in vitro system, you can make also crazy things, like you can, for example, put an atrium actually in the middle of the two ventricles, so between the right ventricle and the left ventricle, and ask the question, is it sufficient to activate both at the same time? And you can see that it is. Now, this is not just calcium signaling. You can do this also with an MEA that is in high resolution, and you see there the same thing. The signal will start from the atrial region, go while the left ventricular region ends in the right ventricular region. Now, I will not have time to go now through all of the applications with this, but I can maybe summarize what we did in this work. So first, we did, we started human pluripotent stem cell differentiation into all these different cardiac progenitors that represent the different progenitors that we have also during cardiac development. They then develop further into the main different compartments of the heart, the atria, AVC region, left ventricle, right ventricle, and outflow tract. We then tested these regions functionally using MEAs, also patch clamp that I didn't show you. Also, we looked at calcium propagation and GCAM, and we looked also at contraction analysis. And we did this both in the presence of drugs, but also on wild types. And we did this also with mutants and teratogenic type of treatments. We also looked at how did these compartments interact, how they started, how do they combine and start essentially a beat, how the contraction starts, and also how certain drug treatments can interfere with this process, with the process of communication between the chambers, which is also something now that is developing, and that is very valuable, because before we couldn't even look at this type of effects. And then we can also look at compartment-specific phenotypes using different knockouts. This is what you saw. I'll show you just one example. I let one, but we looked actually 3 of them, where we essentially validated what we know from in vivo, but we also gained new insights because we used a mutation, for example, FOXF1, that wasn't really studied before. And also we looked at the effects of different teratogens, like citretin that I showed you, but also thalidomide showed a very specific effect, again, that reproduces something that we also saw in, or what you can see in patients treated with thalidomide. Now, what is outgoing, or what is ongoing in the lab, and what is actually the outlook now? So until now, what I showed you was, okay, the very early stages, the lab very much focused on the early stages of cardiac development, human cardiac development, where we first looked at developing left ventricle. That was the first study, and then in this follow-up study, we looked at the development of all the other parts of the heart. But now we want to understand, how does the heart actually start to grow and develop, in the next stage. And that stage basically is what we call the trabecular stage of cardiogenesis. It's very important for fetal growth and injury, but also for regeneration, for fetal regeneration. Another region that is important is another aspect of what is important is coronary vascularization that comes from these sinus vesicolus progenitors that will invade the cardioid, the whole cardioid, and start forming these sort of vessels that you can see here at this stage of development. This is extremely important, as you can imagine. Coronary, to have a cardioid also that starts recapitulating coronary function. And then finally, we have also cardioids that can, interact, where you see here interacting right ventricle and left ventricle. And now, of course, it's interesting to start integrating into the system, for example, pacemakers, start looking at developing septation, and also with all of these aspects here, look at the mechanobiology of cardioids. Because I think this is the actual advantage of the system, because you have this working chamber that starts growing and developing in the context of all of these different tissues. And then with this, I would like to end with the acknowledgements. First, I'd like to, of course, thank the lab. It was really a huge team effort. The first part of the work was led by Pablo, Nora, and Stefan. Then the second study was led by Clara and Alison together with Tobias, but also the whole group actually contributed to this work. Then, of course, all the collaborators that were really invaluable in this. Of course, the different funders. What was especially important here was co-funding, so that we really could do also risky experiments and not just basically grant-based funding. And then we also managed to spin out a company, Heartbeat Bio, a startup from IMBA, that is now developing this platform further, automating it and developing disease models. While what we do in the lab is, as I said, developing this model further to the next stage of cardiac development, as we believe that this is going to be also more physiological, because we are, and also then predictive later on as we develop also disease models based on these more upgraded models. And with this, I would like to thank you, and I'm happy to take questions. Thank you.
Thank you so much, Dr. Mendjan, for that fantastic talk. We do have some questions from our viewers already, so let's get started. Following on the development process, can you elaborate on what happens after these initial stages, and what is the next step in the development of these human cardioids?
Yeah, so I sort of alluded to it a little bit. This is this trabecular stage that we call compaction. So what happens there, essentially, the heart starts to grow from about 0.5 millimeter, you have to imagine. So really, when the human heart starts to beat, we are the embryo is about the size of 2 millimeters, and the heart is about 0.5 millimeter. And then, of course, it has to grow rapidly in size. So that's while it functions at the same time. So it's quite a special organ because it grows and functions while it develops at the same time. And this is the stage that we want to now mimic. So basically this growth, proliferation and growth, and also vascularization of the whole heart, because we think that after that, once you then start maturing a structure that can grow further, we will get something much more functional and something much more useful also for the clinical and the pharmacological community. Yeah.
Fantastic. So within the platform, can these cardioids continue to grow and mature, or is there a limit to their development?
They can actually be left in culture for months. So because they are quite, very sort of clean, there are no contaminating cell types there. So they're really just cardiac cell types there. They exit, as also cardiomyocytes do in vivo, they exit their cell cycle. And the cardiomyocytes, they will just stay cardiomyocytes and then, slowly mature. And of course, we can also mature them metabolically. But as I said, we cannot just simply jump development. We need to also develop now, the next stage of the developing system and then mature how it really happens during development.
And do you see septation?
No, we didn't see that yet. This is something that we want to start working on because, septation defects are the most common defects. The actual problem already starts in the progenitors. We know that. But then, of course, a septation defect will be the final, sort of manifestation of that early molecular defect. So that's why this would be a very useful model then, of course, also to start studying that. So yeah, it's something we want to do.
Thank you. So while the focus is on development, can this technology using cardioids be used to study interventions such as pacemakers to treat heart defects?
Yeah, I think so. But I think that to what extent this platform can be used, I think this still needs to be determined. This is something that we need to test. Also, Heartbeat Bio, I think this is what they are mainly now focused on to see, which conditions can be modeled better and not. I think arrhythmias in general in the field are known to be actually can be modeled quite well using human pluripotent stem cell cardiomyocytes and also using cardioids. The reason is because these early stage developmental cardiomyocytes, they're quite similar to the pacemakers in terms of, some of the active physiological properties, and they react quite similar also to drugs. So they're very useful and already way more predictive than, for example, some of the animal models. So this is there in the arrhythmic field is already they're very useful. And now, of course, because we are adding special cardiomyocytes like the atrial cardiomyocytes, etc., this is becoming even more interesting. Yeah.
Thank you. So we do have a question that just came up from a viewer. So you highlighted the use of microfluidics to mimic blood flow. Can you elaborate on the specific challenges of integrating microfluidic systems with these complex 3D human cardioid models?
Yeah. So microfluidics, of course, would be something very interesting to start, to start combining like this type of organoid system what we have here with the microfluidic platform. And the main sort of challenge will be that this platform is high throughput and the microfluidic systems usually are not, or they are quite difficult to, or will be also very cost prohibitive, right, to develop them at a large scale so they can be really used also at the industrial scale, for example. And I think this is where I see the biggest challenge, to bring these engineering technologies together with this sort of self-organizing technologies, organoid technologies, but in a way that it's not too elaborate and too expensive and too complex. Because the more complex a system, the more difficult it will be then to analyze and to also understand what is going on.
Fantastic. Thank you for answering those questions. As we draw to a close, we do have some time to answer just one final question. So the potential for drug discovery using these cardioid models seems very promising. Can you elaborate on how this platform might be used to screen for new drugs or test the efficacy of existing ones?
Yeah, I think one aspect that can be already done now is to look at, for example, the toxicology or the toxic effects of any drug, right? Because any drug that we need to would come to a market needs to be tested for its toxic effects on the development of the embryo, to be essentially sure can it really be given also, for example, to pregnant women, but also in general, the toxicology, for example, doxorubicin and these type of toxicologies, right, like anti-cancer drugs, one could already start using this type of model to look at that. Furthermore, what I mentioned, another area are the arrhythmias. This is, I think, where this model now, I think, is also quite far advanced, that you can also start looking at very specific arrhythmias that will affect specific things. Then another area are also, of course, congenital defects that will happen, that will affect certain compartments, and especially also drugs that will help manage these. Because you can imagine that people with a congenital defect, it's not just that they have a defect, a birth defect, but they continue to have all sorts of other cardiovascular problems throughout, and that, of course, we need a model for that. And I think the biggest potential I see as we start further maturing and developing this platform in a growing heart, that then can, of course, start modeling also more adult type of diseases, which means also you can have a platform to study drugs that will affect adult stages. Although I think that some aspects you can probably already do it at the earlier stage, because, for example, the mechanical aspects of cardioids, I think they are a big advantage, because you start modeling aspects of the biology, of cardiac biology there, that are universally important, both in development, but also later in adults and in disease.
That's great. Thank you so much for answering all those great questions. Unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be answered offline as soon as possible. So that's it for this year's Innovations in Disease Modeling Symposium. As mentioned throughout the day, all the talks from today's event will be made available to view on demand for the next 30 days. You can access the talks via the same link you used to access them today, and we will be sure to pass on any further questions you submit through the system to the relevant speaker. That just leaves me to say thanks again to Dr. Sasha Mendjan and all the amazing speakers for these past few days for sharing their research with us. And to all our viewers, we will see you again at our next online symposium, The Landscape of Cancer Research, Advances in Immuno-Oncology. Thank you and goodbye.